Welcome back to cell biology. We are in the middle of our cell cycle chapter. We've talked about the steps of the cell cycle in our first video. We talked about the role of microtubules. Now we're going to talk about that process of cytokinesis. Cytokinesis is the process of dividing the cytoplasm. So not only do we have to copy our DNA, make sure our two daughter cells are going to have complete copies of their genome, have functional nuclei. We also have to make sure that they have the liquidy, gooey stuff in it and their appropriate organelles. So once chromosomes have separated, next we can have cytokinesis, which is going to divide the cytoplasm in half or into two. Usually this can start as early as late anaphase or even during early telophase. Sometimes it follows telophase after that step. Some cells are able to go through multiple rounds of mitosis, multiple rounds of nuclear division without going through cytokinesis. And that cell that has multiple nuclei in it is going to be called a syncytia. Sometimes this is a permanent part of their division. Sometimes this is a temporary part of their division. Sometimes this is something that is going to be seen by an infectious particle. So sometimes the existence of syncytia can tell you there's been a viral infection. In an animal cell, cytoplasmic division is called cleavage. Cleavage is going to start by forming this little cleavage furrow here, this puckering in the cell. It's like you're taking a shoelace and just circling it around it and gradually pulling it tighter, or like pulling the drawstrings on sweatpants or something. That furrow continues to deepen as you're pulling that waistband, the drawstring, as you're pulling it tighter and tighter until the two ends are in contact with each other and they're able to separate. Except here, this waistband isn't like a shoelace or a string. This is a ring made of actin, which is a protein involved in giving your cells shape. This furrow is going to occur at the spindle equator. That means it's the midpoint between where our two spindles were. And so if your spindles are on opposite ends of the cell, you end up with two daughter cells that are about the same size. So the spindle midzone is that central part of between your two spindles. And your cell can figure out where that spindle midzone is because each spindle is sending out signals. In animals like nematodes and fruit flies, that signal would come from central spindlin that gives a signal to create that spindle midzone. And that is a required part for them to be able to complete the steps of cytokinesis. Cleavage is going to depend on the ring of actin filaments, or the contractile ring, which is what I was just comparing to the drawstring in your sweatpants. So this contractile ring of actin here in the midpoint between our spindles, it's going to form a ring around this cell right below the plasma membrane. It starts forming that ring late or early in anaphase, and it's going to keep tightening as cleavage progresses. Here you can see Rho being activated, leading to where we're going to form the cleavage furrow. Here you can see the mid-body between these cells. The ring gets the energy to contract through the interactions of actin and non-muscle myosin 2. Non-muscle myosin 2 is a type of motor protein. Rho GTP binding proteins are going to regulate how those proteins are assembling together and how they're being activated. And Rho A, which we just saw here, Rho A is going to be recruited to the cleavage furrow, and it will start activating the proteins that allow actin to polymerize. So that is what forms the ring. It also is going to stimulate the activation of myosin, which is the motor protein. As cytokinesis ends, that connection between the two cells is going to narrow down to a thin stalk, and that's going to hang around for a little bit of time. In the middle of that stalk is the mid-body. Midbody is pretty electron dense. It's got microtubules left over from the breakdown of your spindle. And that midbody persists until the active abscission. You might be familiar with that term abscission for being the word of how leaves fall from trees. So it's how the things break off. Here it's still breaking off, but it's two cells breaking off from each other. So at the end of cytokinesis, abscission is that step of two daughter cells finally separating from each other. Plant cells also go through cytokinesis, but they do it differently. And we're just talking about plant cells because they're interesting, because it's weird to see how they're different. But this is a class focused on mammals, so you don't need to worry about, memorizing these things or studying. I'm not going to ask you to tell me about plant cells on quizzes or exams, but it's still interesting to learn about. So plants can't form that contractile ring. You can't just, squeeze this ring of actin filaments and pinch them in half, because they have this cell wall, and that cell wall is pretty sturdy. So instead, they start making little pieces of plasma membrane and cell walls in these little vesicles that line up between the two nuclei. During late anaphase or early prophase, the Golgi vesicles are going to start making the Golgi is going to start making the vesicles, and they are able to start lining up between the poles. Then these little vesicles are going to start merging with each other to form a cell plate, which is got cell membrane and cell wall inside of it. And eventually that spreads to contact the cell wall and cell membrane and divide that cell in half. The vesicles, which have the polysaccharides and glycoproteins that are involved in the cell wall, will move to the spindle equated by the phragmoplast. The phragmoplast is an array of microtubules that are deriving from the polar microtubules, and those vesicles fuse together to form a cell plate, which expands outward towards the cell wall. Some animals or some organisms are able to do asymmetric cell division. So instead of having two daughter cells that are about the same size, that asymmetry means you're going to end up with cells that are about different sizes. So one example would be in yeast that bud. They end up with one large and one small cell. We also see a lot of these asymmetric divisions during embryonic development that leads to a frog embryo that looks like this, where we have smaller cells and larger cells. Or in the sea urchin, you have a larger cell and a smaller cell forming. That's because our spindles are not totally at different poles. You have this one at the pole and this one here. So the midpoint between them is here. So you're going to end up with your actin ring here and a small cell and a large cell. In bacteria, cytokinesis usually involves the cytoskeletal protein FITZ. FITZ is pretty similar to tubulin. It is still a protein involved in, giving structure and shape to the cell. FITZ will form a ring here at the midpoint of the cell, so the two daughter cells can pinch off. And again, this class is not focusing on bacteria, but it is relevant to our cells because we have mitochondria that are going to act like bacteria. So they're going to do cytokinesis, very similar to how bacteria are doing binary fission. All right, that is how cytokinesis behaves in different types of organisms. We're going to come talk about how we are regulating the cell cycle, how a cell knows if it is safe to go through it, how it knows when and how to go through those different steps of the cell cycle. That's what we'll talk on in our next video. I'll see you then. Bye.